So thanks very much for the organizers for inviting me. I'm given the collection of people that are at this meeting. I'm actually very, very sorry that I cannot meet you in person. Since we're among physicists, mostly in the audience, I thought it was maybe a good idea to start by explaining you why I'm not working on the things that I'm showing on the first slide here. So this is the Lagranglan of the standard model and general relativity field equations. And if you want, from the point of view of a theoretical physics, you could say, well, we're sort of done explaining everything that happens in the universe, because if you just start hot and small with a big bang and you simply iterate these equations forward in time, then you should get our universe. And this always struck me as somehow really missing the key point. So for example, I see nothing here on this slide that would explain to me why, if I were to do this, I would get things like, for example, this or that, or even this for that matter. So at the sort of a very fundamental level, what I'm interested in is understanding how, from the fundamental physical laws, you get these very, very interesting, beautiful, and complex behaviors emerge. And so the area that I've chosen myself to sort of focus a lot of my research on is gene regulatory networks. And the reason is that gene regulatory networks are sort of responsible for many of the very complex behaviors that are unique to life. So one thing in which living things are really fundamentally different from dead things is that they can respond on their own behalf to their environment. So for example, this bacterium that you see here, as the environment changes, it can change its internal state in such a way as to now allow it to let's say, eat the orange nutrient and grow. And in that sense, you can really talk about this bacterium as responding on its own behalf to this change in the environment. They also allow to express one genotype into this beautiful array of phenotypes that allow these sort of multicellular societies of cells to emerge. And what is nice from the point of view of a physicist is that we sort of understand the basic physical mechanisms involved. In the end, it's all encoded in small DNA sequence fragments in the DNA that are bound in a sequence-specific manner by regulatory factors. All right, so for today, I'm going to focus on stochastic gene regulation in bacteria. And so the agenda of the talk, in the first half, I want to talk to you about what determines gene expression noise across genes and conditions genome-wide in E. coli, and what we think we've learned about that. And the second part is going to be about the extent to which regulatory responses in bacteria are context dependent. They're dependent on how they depend on the state of the cell at the time that the signal comes. All right. So to set up the general topic, I want to start from what is sort of the most simple null model of how gene expression works in bacteria. And this model assumes that the following processes basically take place at constant rates. So there is transcription of the gene into mRNA, the decay of the mRNA, translation of the mRNA into proteins, decay of proteins, and exponential growth of the cell. Now, first thing I would like to note is that for many proteins, the dilution typically dominates protein decay. All right, so the proteins are stable over typical multiple cell cycles, which means that the actual concentrations of proteins are a balance between production and dilution. So with this very simple null model, the mean number of proteins per unit volume is given by this product of rates, and the intrinsic noise in protein concentrations is going to be given essentially by Poisson statistics, and so it's going to scale inversely proportional with the mean expression of the protein. All right, so what does this actually lead to in practice? Now, the first thing to note is that this intrinsic noise is really not negligible. For the large majority of genes in the E. coli genome, the average number of mRNAs per cell is less than 1. Okay, so most of the time when you look for a particular gene, you will not find one of its mRNAs. And even at the protein level, the large majority of genes have less than 100 proteins per cell on average. So this means that there is these Poisson fluctuations, they're significant. You cannot really neglect these. But on top of that, of course, there are many other sources of noise. So in particularly, the model assumes all these rates are constant, whereas in reality, of course, these rates are fluctuating in time and from cell to cell in a way that we currently really don't know that much about. And in particular, what's important is that the rate of transcription of the gene will depend on the state of the promoter. And so this rate will fluctuate in time as transcription factors in the cell are binding and unbinding from this promoter. This transcription rate of the promoter will switch between different rates. All right, so that's the setup. So the questions that we were interested to look at is, how does transcriptional noise vary across genes genome-wide? Are they basically equally noisy? Are there big changes? What determines these changes? How do these noise levels vary across conditions for the same gene? And is it possible to predict how noisy a gene is going to be in its gene expression from its sequence? So to do that, we made use of a library of transcriptional reporters made in the lab of Erik Van Nimwegen, where basically every promoter region in the E. coli genome was fused in front of GFP on a low copy number plasmid. And so transforming these plasmids into cells, you basically have transcriptional reporters for every promoter region in E. coli. And then you can, for example, use flow cytometry to, in high throughput, measure single cell expression levels for all promoters for thousands of genes. So if you do that, you find for almost all genes that the expression levels across cells look roughly log normal. So it can be approximated by a normal distribution of log expression levels. And so you can characterize the expression of each promoter by giving mean and variance in log expression. The first thing that we found that was actually quite surprising to us is that it seems that low noise is the default behavior of E. coli promoters. So in this plot, every black dot is an E. coli promoter, and on the horizontal axis is its mean level, and on the vertical axis is its noise level of this promoter. And now we compare these noise levels with noise levels of synthetic promoters that we made in the lab. And what these promoters are essentially just roughly 100 base pair long, completely random sequence fragments of DNA that we screened for driving expression. Okay, so from a very large library, we pulled out all the sequences that drive expression. And in red, you see what the noise levels are of these synthetic random sequences. And what you see is that many native promoters are much more noisy than these minimal synthetic promoters. So if you make just a random sequence that expresses, it's going to be low noise by default, but there are many native promoters that are much more noisy than that. So this raises the question, what determines whether a native promoter is noisy or not? And so to make a long story short, what we found is that the more regulated the promoter is, the more noisy it tends to be. Okay, so you can use computational and experimental annotations of which regulators regulate which genes in E. coli. And then what you see is as you sort your genes from lowest noise to highest noise, and you calculate the average number of different regulatory inputs per gene, you find that as you go from low noise genes to high noise genes, the average number of regulatory inputs per gene systematically goes up. Okay, so unregulated genes are low noise, and that includes our synthetic promoters, whereas the more regulated the promoter gets, the more noisy it gets. So this was done in one condition in glucose, but we've since gone and measured gene expression noise across 9 different conditions for all genes, genome-wide. And this includes different nutrients and also different stresses and stationary phase. And we find that this relationship holds across all growth conditions. More regulated genes are more noisy than unregulated genes. However, if you look at individual genes, you find that not only the mean expression level of the gene, but also its noise level varies significantly from promoter to promoter. Okay, so the noise level of a promoter is not an intrinsic property of that promoter, but actually depends on the growth condition. And if you go from one growth condition to another, a promoter that was very low noise in one condition may become high noise in another condition, and vice versa. So we think that the explanation for this behavior is that the differences in noise levels of different E. coli promoters is driven by propagation of noise from regulators to their targets. So in each condition, each transcription factor will itself have some level of noise, all right? So the transcription factor's concentration will fluctuate from cell to cell in some way, and some transcription factors may have less noise, and other transcription factors like this pink one may have more noise. And the noise level of a gene is now going to be a reflection of the noise levels of the regulators that regulate it in that condition. So for example, in this condition, this gene would have lower noise, the gene regulated by the pink transcription factor would have higher noise, and the gene regulated by both would still have higher noise. So you would get this dependence that the more regulatory inputs you have, the more likely you are to be noisy. But how noisy you are, it depends on which transcription factors are more or less noisy in each condition. So the noise levels are really determined by how noise propagates through the regulatory network. So to sort of check if this interpretation is reasonable, one thing that we did is we said, well, then we should be able to explain some variation in noise levels by a very simple naive model that models the noise of a given promoter P in a condition C as basically a linear combination of the number of binding sites that that promoter has for each regulator T, for each transcription factor T, times how much this transcription factor T propagates noise in each condition A. And so these noise propagation activities here in blue, we fit those so as to explain the noise levels of the target promoters. And we find that we can fit up to 30% of the variance in noise levels across genes by such a very naive model. And this you cannot do if you randomize the association between what sites are in promoters and what are the noise levels. And so in particular, this model then also allows you to work out in each condition what are transcription factors that are propagating most noise, either in specific conditions or across all conditions. And so in particular, we found a number of transcription factors that seem to be significantly propagating noise in all conditions. So targets of these promoters in all these growth conditions tend to become more noisy. All right. So now we thus believe that in any one given condition, the relative levels of noise of different genes are basically set by how the noise propagates to the regulatory process. And so in each condition, unregulated promoters are always the least noisy. However, the absolute value of this noise floor that is exhibited by unregulated promoters we noticed varies across condition. All right, so in different conditions, the lower bound on noise that is sort of expressed by the unregulated promoters is different. And what we noticed is that there is actually a very good correlation between the growth rate in each condition and this noise level and this noise floor, the lowest level of the noise. So there is some basal level of noise that all promoters have, and this depends directly on the growth rate. When cells are growing slowly, all the noise levels tend to be high. And when cells are growing fast, these noise levels go down. So this raises the question of why would it be that you have this systematic decrease of noise with growth rate? And this brings me now to the second part of the talk. And in this part, we were wondering about the question of, is it possible for bacteria to respond to regulatory systems in a context-dependent way? So as you all know, organisms with brains respond to signals in a very context-dependent manner. So for example, this orange dot is the same size as this one. This square is the same color as that square. And you also are probably aware of the fact that different people have different ideas about what the color of this dress is. So the question is, do such context-dependent responses also occur in bacteria? And if so, what kind of regulatory network would implement this? And what kind of variables are key for setting the context? So I will already tell you the answer. And the answer that we found is that the actual current growth rate of a cell is a key determinant of its sensitivity to signals from the environment. And the more slowly a cell grows, the more sensitive it becomes to signals from its environment. And this happens automatically without the need of any regulatory circuitry. Okay, so why is this? So we've been in the lab now for a number of years setting up an integrated system of microfluidics, time-lapse microscopy, and automated image analysis that allows us to track responses in single bacterial cells, regulatory responses, to varying growth conditions. So this is a variant of the so-called mother machine, where cells are growing in these narrow 1D channels, growing in single file. And we extended this design by having basically allowing for two inputs that with programmable pressure pumps, you can vary in a controlled way in time. And so this allows us to expose cells to different media in a controlled way and track their responses in real time with time-lapse microscopy. Okay, so one of the first systems that we looked at is sort of like the hydrogen atom of gene regulation. This is the lacOperon. Probably most of you are familiar. So there's a set of genes, lacZ, which is for metabolizing lactose. lacY is a transporter of lactose. So when lactose is not present, this regulator lacI represses the expression of the lacOperon. When lactose comes in the environment, and it's transported into the cell, it will be turned by lacZ into allolactose. This binds to lacI. This then releases the repression of the promoter, and the gene switches on. So now in the movie I'm going to show you, we're growing cells in this device and switching between glucose and lactose every 4 hours. And what you will see is that after 4 hours, cells will go into growth arrest and then sit there for a stochastic period of time until the cells start inducing the lacOperon and recommence growth. So now they stop, and now you see that various cells start growing, whereas others are still not growing yet. And so there is some stochastic distribution of waiting time. Once you go back into glucose, the lactose is diluted out, you see it disappearing, and now upon subsequent switches, we no longer see growth arrest. Okay, so this only happens in the later switches. The cells have memory of that they've seen that lactose. But the thing I want to focus on today is this distribution of lag time. So we measured the distribution of lags in single cells and found surprisingly that it's bimodal. So about 1/3 of cells, 1/4 to 1/3 of cells, they respond fast within 20 to 45 minutes. 2/3 of cells take much longer, somewhere between 50 minutes and 3 hours, and a small fraction of cells we never see respond. And to make a long story short, we found out that what determines these lags has something to do with pre-existing levels of lactose, of operon expression, at the switch. So in particular, by fiddling a little bit with the expression of this lacY repressor, we can change the amount of pre-existing lac expression before lactose comes in the environment. And we can show that we can systematically shift around this distribution of single cell lags by tweaking the expression of this lacY protein. So we know that somehow whether a cell is going to respond fast or slow depends on how much pre-existing lac expression that cell had. So we went and checked how much pre-existing lac operon expression do you need to kick in this positive feedback loop and to express and start expressing the lac operon at a high level. And to do this, we did a sort of a dilution experiment where we induced the cells in lactose, and then we switch them to glucose, and we dilute out the lac operon expression for different periods of time, and then go back to lactose and ask which cells now switch quick and which cells switch slow, and compare it with how much lactose they had at the end of the previous lac phase, and how much have they diluted it out. And what we found from that is that basically these fast cells are all cells that have non-zero expression of lacY and lacZ. All right, so as soon as you have 1, 2, 3 molecules of lac operon expression, that's enough to kick in the positive feedback loop and be fast. And the only cells that are slow are cells that have 0 lac expression, which makes them effectively blind for the lactose that comes in the environment, and therefore that you have to wait until they have a stochastic burst of lac expression before they switch on. So this is very nice, but it was also quite surprising to us because there is previous work from the group of lacY that has suggested, also in single cell experiments, that to turn on the positive feedback of the lac operon, you need like hundreds of molecules of lac expression. So the question is, what's the key difference between those experiments and ours? Now, those experiments are done with an artificial inducer, synthetic inducer. And in those experiments, the cells grow in glycerol and they keep growing as they're being induced. Whereas in our case, cells go transiently into growth arrest. So this made us wonder, what is actually the effect on the circuit of cells going into growth arrest? And so we took some very simple model, just some simple coupled differential equation model. It's an adaptation of models that have been used previously for this. But so to make a long story short, what we find is that very, very simple modeling explains that the critical level of an inducer in the environment drops with the growth rate of the cell, increases with the growth rate of the cell. All right, so, and you can easily understand why this is the case. So the positive feedback loop is triggered when you reach a certain critical amount of intracellular inducer. But the intracellular amount of inducer is given by a balance of influx through the transporter and dilution due to growth. So the faster a cell is growing, the more dilution it is, there will be, the harder it will be to reach a certain critical level of intracellular inducer. And moreover, the concentration of the transporter, which sets the input rate, is itself also going down as a function of the dilution rate. Because again, the level of the transporter is a balance between production of the transporter and dilution. And so together these things conspire to make the actual inducer level grow quadratically with growth rate. Okay, so this is a non-trivial effect. As you increase the doubling time of the cell, the critical level of the inducer drops quadratically. So we decided to check if we can confirm this is the case. So we did some experiments that actually Jacob and Monod could have already done. But as far as we can tell, nobody ever thought of checking this systematically. So basically you do a bulk experiment, and we're going to use this synthetic TMG inducer. And we're going to see, as a function of the concentration of this inducer, when do the cells switch on in lac expression. But now, instead of doing it in just one condition, we're doing it in different growth conditions that systematically vary the growth rate. Okay, so the growth is slowest in pyruvate and it's fastest in arabinose with amino acids added. And so you can ask, what is the critical level in these different growth conditions? And we find that indeed the critical level of the inducer that you need to turn on the lac operon depends crucially on the growth rate of the cell. And in fact, we find that it grows even faster than quadratic. It's almost third power. And so the critical inducer level drops dramatically with dropping time. So I just want to point out that this mechanism is going to affect almost any regulatory switch. Okay, so you can even think of a system that where there is really no inducer whatsoever. You can think of a gene that makes a transcription factor that positively feeds back on its own expression. And whether this positive feedback loop is going to be switched on or off is going to be only a function of growth rate. Okay, and you can, for example, imagine that maybe a phage could make use of such a circuit where, as long as its host cell is growing fast, it will not activate its lysis program. But as soon as the growth rate falls too low, the positive feedback will kick in and will activate. And in fact, there are many regulatory switches in bacteria that are essentially one of these positive feedbacks, operon feeding positively on its own expression, that is coupled to a signal. So this is competence in Bacillus subtilis, sporulation is a similar case, and many two-component systems are also such that there is a positive feedback loop that is coupled to a signal that, in, for example, this case, control the dimerization. So in all these cases, you get phase diagrams where the response of the system will not only depend on the level of the inducer, but also critically on the growth rate of the system. So to summarize what I've talked about, fast-growing cells effectively don't listen to signals of the environment. They effectively just mute these signals and ignore what's going on, which maybe makes sense given the fact that they're growing nicely. However, when cells start growing slow or go into growth arrest, they become very, very sensitive to even small fluctuations of signals in their environments. And we believe that this may also explain why growing cells are generally much more heterogeneous than, why growth arrested cells are more heterogeneous than fast growing cells. And second, in the first half, I showed you that the noise of gene expression in bacteria can be decomposed in two components. There's a noise floor that depends on, that does not depend on the promoter, but decreases with growth rate. And then there is a promoter dependence noise propagation component that is really a feature of how noise propagates to the regulatory network. And noise is therefore itself a regulated property that's encoded in the regulation of each promoter and is therefore highly context dependent. All right. Now, I will basically stop here. I will just tell you that you can ask, is there any use for this noise propagation? And so we've developed a theory that I don't have time to explain to you now that shows that very generally, noise propagation is a positive feature for these bacterial regulatory systems, and that effectively allow cells to have a continuum of regulatory strategies that interpolate by complete bet-hedging where all that a regulator does is make its target genes more noisy, up to very highly correlated precise regulation, but with any intermediate in between, where regulation is a combination of regulators pushing the targets roughly in the right condition and noise propagation, adding noise to this to add a bet-hedging aspect to this regulation. All right, so with that, I've come to the end. I just want to show you the people that have worked on these things. So the original synthetic promoters were evolved by Louise together with Olin in my lab. This dual input mother machine was set up by Matthias, and we collaborated with the group of Jean Myers and Florian Hug on the automated image analysis. And then the two stories I told you about today were one work of Arantxa Urtxu-Eguea, who was a PhD student in the lab, and Thomas Joulou, who is actually in charge of all the wet lab experiments that happen in my lab. All right, so with that, thanks for your attention. I'd be happy to answer any questions.
Yeah, thank you for this nice talk. So we have a few minutes for questions. So there's a question by Ayan.
Yeah. Hello, Professor Nimwegen.
Hi.
That was a wonderful talk. So I was just wondering, so you have mentioned this promoter independent noise floor and then promoter dependent this propagation of noise, no? So can it be compared with the intrinsic and extrinsic component of noise? And when I say extrinsic component, I mean the noise due to the sharing of similar resources like RNAP or ribosome and etc., where this intrinsic is basically the biochemical noise due to the low copy number. Can it be compared?
Well, yeah, you can do that, but I wouldn't do that. So there is intrinsic noise because the process is just a Poisson process, right? So that only the rates are fixed, and so there will, even if all the rates are constant, there will be Poisson noise. Then on top of that is what you're saying. So the noise floor is also determined by fluctuations in polymerase levels, ribosome levels, RNases, fluctuations in the growth rate itself, right? Because single cells don't grow always at the same rate. This also fluctuates. This is actually how much each of these components contributes to this noise floor is something we're working on in the lab and trying to work out precisely. And all I can tell you is that it's not trivial. Yes, it probably has something to do with global components, but how exactly, I think at the moment we don't know.
Thank you.
Thank you. So there is a question by Thierry or Alexandra. I don't know which one.
Yeah, it's me. Hi. Yeah, great talk. I had a question. So if I understand your reasoning, you're thinking that when the growth rate decreases, you go from the non-induced to the bistable phase, which it explains the increasing noise. Could you imagine the other way around, like going from, as the growth rate decreases, you go from the bistable phase to the always induced phase, in which case you would actually reduce the noise by reducing the growth rate?
Yes. Yeah, I think you can tune your system to be either in one or the other regime.
Right. So you're saying that the claim is that the system is actually wired in such a way that these, inducible, bistable systems actually by default, when you have a high growth rate, they're actually in the non-induced phase, and then they start listening to the environment. But do you have, except for this, for the lacopron, do you have, have you thought about a way to test this more generally across all genes?
Well, so the only thing that I can tell you right now, and this is in the paper of Thomas that just came out, is that we looked at all two component systems for which proteomics data is available across a bunch of conditions. And a significant number of them are in a regime where you would expect a whole bunch of these guys to be at really zero expression. Okay, so it's generally the case that these things are very low expressed and really need to be induced to get going.
And so if I may follow up, like, did you try to write down some simple model that would actually explain this noise floor dependency based on what on the story you just said?
Yeah, but I promise you that I would have published it if it was really working according to what the simple model said. So yeah, no, I mean, the people are doing experiments in the lab to figure out how these things scale as you change these growth conditions and so on. And it's just all a little bit more involved than we originally thought.
Okay.
So I have a quick question.
Yeah.
Do you understand why some transcription factors are always very noisy and what's so special about them in all conditions, the one that have high noise in all conditions?
Yeah, no, okay, so there is a couple of things that you could say about this. So one thing you could say is, well, these are just things that have lots of targets, which is true for things like HNS, sigma 38 CRP. But it's, for example, not true for this GAT-X, GAT-W, which have to do with acid stress. The other thing is that you could say is maybe this is just special to growing cells in microtiter wells in the way that we're doing, right? So all these experiments, even though there are different growth conditions, all these guys are, you know, we have 96 plates, and maybe, yeah, this is where acid stress is an issue that starts coming up halfway through your exponential growth phase. And so I don't know whether there is something deep to this, or this is something that has more to do with the kind of conditions we can easily do in the lab.
Thank you.
And do you think, so just to continue on that, do you think that the fact that some transcription factors should form some complex, so many of them act as dimer or tetramer, or maybe there may be cooperativity between the binding sites? Do you think this could also maybe challenge the linear model, which is only based on the number of binding sites?
Yeah, the linear model is terrible. No, I mean, it's not intended to be any realistic picture of what's going on. It's only to sort of show that let's say within one condition, there is a correlation in which genes become more noisy, and this correlation has something to do with which genes share binding sites for the same transcription factor. And right now, that's really all it says. We don't have a realistic model. And I think in reality, this is actually, if you wanted to be accurate and predict the actual noise level, let's say within 50% of the true value, you would need really a very different model.
Yeah, okay. I think there were questions, but it disappeared. So then we can move on. So thank you again for this nice presentation. So, so our next speaker is